In this lecture, we're going to dig into some of the concepts with sugar and sugar metabolism, looking at the metabolites as they flow through our different metabolic processes. I want to start with just a review of the different types of carbohydrates and the different forms that they'll take. So one of the most common, that one of the foundational pieces of our metabolic processes is this glucose molecule. So this is called an aldopentose because there's an aldehyde functional group. We have 1, 2, 3, 4, 5 carbons in that group, or 6, excuse me. So the hexane or the hexose part, they are the OSE because they're sugars. We have hydroxide groups on every one of the carbons besides the aldehyde. So we have 6 for the hex, the aldehyde on top, and that's why it's called an aldohexose. This is the monomer for starch and cellulose, so for most components of carbohydrates. We'll also see ribose. It is an aldo, right? An aldehyde, and 1, 2, 3, 4, 5 carbons in its backbone for pentane. But it's a OSE for the sugar because each of the carbons has a hydroxide group on it. It's a component of ATP, various coenzymes, and RNA. Finally, we have fructose. It's a ketohexose. You've guessed it. The keto is for the ketone here because it's in the middle of the chain rather than the end. It's a 6-carbon chain for hex, and each of the carbons has a hydroxide where it doesn't have this carbonyl group on it. This is found in corn syrup, different types of fruits. It's also a very sweet compound. These are some of the main starting points for metabolism. Where we have ribose and fructose, they can be fairly easily converted into glucose. And those would be really, ultimately, we're going to focus on glucose, but we can generate glucose from both of these if we need to. I want you to take a second to think about this based off of what I just told you about those different types of sugars, and pause and then try to answer the question, and then we'll continue on. All right, so the solution here, the first step is to determine if this monosaccharide is an aldose or a ketose, and then determine the number of carbons present. So this one, we can see at the end, we have the carbonyl group on the end. That means it's an aldehyde, so that would be an aldose sugar. And it contains 6 carbons, so it's going to be a hexose. So this will be an aldohexose. These different sugars do have chirality, which means that there are 4 different unique groups attached. We can see with D-glyceraldehyde, it has a hydrogen, hydroxide, a carbonyl group, and an alcohol. So 4 different things. So this is one of its chiral centers. And if you recall from organic chemistry, we want to get things on the bottom. That means it's in the back. So high priority group here is the oxygen, then the carbonyl group, then the hydrogen is actually 4th, and so this is 1, 2, 3, and 4. So in order to name this, so it'd be a right-handed group because we'd have to swap these out. We're not going to worry about the naming system, but just recognize that this is a mirror image of this group. So these two are enantiomers of each other. They're mirror images. If I have an enzyme that recognizes D-glyceraldehyde, that enzyme is not going to recognize L-glyceraldehyde because the shapes are different and they won't be able to recognize these different chiral centers. So I'm not going to make you name these. I just wanted to talk about this so you are aware in case you come across this later. Usually in terms of these are kind of in these Fisher projections, and you kind of have to imagine that the red bonds to the right and the left are coming kind of at you because of the shape. This is a 3-dimensional shape, so we kind of see this carbon right here is going to be at the intersection, and the bonds going away from you are going to be in the red, and you can kind of see how this swaps kind of back and forth. But we're supposed to imagine these going down into the page and these coming out at you. And generally, by convention, the oxidized carbon, the carbon that's on the the aldehyde or the ketone is placed on the top of the compound. And if the OH on the bottom is on the right, it's generally called a D. If it's on the left, it's called an L. This is a little less sophisticated than some of the naming procedures that we had in organic chemistry. That's why I'm not going to have you generally name these. But the big picture thing I do want you to recognize is that these are mirror images of each other. And because of that, there's only one enzyme that's going to regulate that. Now notice this isn't shown as a mirror image. We would need to push this hydroxide over here for it to be mirror image. But remember, this can freely rotate. This isn't a chiral center because it's attached to two hydrogens, which are the same thing. So anytime you have only three different unique things and two of them match, it's not a chiral center. So we don't have to worry about the orientation of this. But this orientation, the hydroxide and hydrogen, because we have a carbonyl, an alcohol, and these two other groups, there's four unique centers, and so this middle carbon here at the intersection is a chiral center. All right, so you may be wondering why we generally show you the carbohydrates as a hexagonal ring. So if you'll go back to this first slide, right, we have this is our sugar. Why is it like that? And now you're showing me stuff that's a straight chain. So basically what's happening is, you notice that I mentioned there's an aldehyde or there's a ketone on the top. So that is what this group is going to be, the aldehyde. And I also mentioned there are a bunch of alcohol groups, functional groups off of each of the carbons. What's happening is that we're going to form a circular ring through this type of chemical reaction. So we're going to have an aldehyde, an alcohol, and then we're going to create a hemiacetyl group. So this is the acetyl group. It's a half of an acetyl group because this is an OR versus an OH. If it was a full acetyl group, it would be an OR up here too. So you have ROCOR would be an acetyl. It's a hemi, which means half, because this is just still a hydroxide. So let's look at this chemical reaction a little closer when we're talking about a glucose molecule. So here is our aldehyde. Here is the alcohol, right? So this is the aldehyde, the alcohol of this reaction. And what happens is this is the open Fisher projection. If we turn it and reorient it so we can see these numbers 1 through 6, and this is the aldehyde, and this is the 6th carbon. This is now the new orientation. We're going to flop this up just so that we can create a more ring-like structure. And that is kind of how we see this. So this 6th carbon is up. This 5th carbon is the hydroxide that's going to actually interact here. And this is then basically the electrons from this oxygen are going to attack this carbon center. The electrons from that double bond are going to pop up to the hydroxide group. Then that forms a bond here, and the aldehyde will turn into a hydroxide group. And if we can generate either beta D-glucose or alpha D-glucose, depending on how that attacks and how that hydroxide forms on the end. So this can be on the up or the down version of this, and that's going to give us two different forms. So these are isomers of each other. These hemiacetyls are very stable, and most of the time the most stable is this beta D-glucose because of where this is on the ring. It's going to be able to be more equatorial on the chain and allow it to be a little more stable. So this doesn't show it very well, but we deal with that stuff in organic chemistry. We're not going to talk about it now, but let's just say the most stable form is beta D-glucose. This is about 60% of glucose in our bodies are going to be in this form, 36 in this form, and a very tiny small amount is going to be the open chain glucose. There are differences in where these hydroxide groups are. So if they're on the bottom or the top of the chain, you can see this previous slide. We have the down, down, up, up, and here we have down, up, or we have up, down, right? So that orientation is going to dictate what different type of sugar it is. So it's just how these are oriented at the different chiral centers. And that's important. Remember, the chiral centers are going to be a different shape. And so different enzymes are going to recognize galactose versus glucose, for example. For a lot of these monomers, our bodies have enzymes that can convert from one to another, specifically to glucose, because glucose is that starting point for glycolysis. Here's fructose, and here it's ketose. And so rather than an aldohexose, so we're going to join a 5-membered ring here instead of 6-membered. And you can see the hydroxide that's going to attack that carbonyl center. So beta D ribose and beta D2 deoxyribose. So the deoxy, right, is this hydroxide is gone. The deoxy removed essentially. These are both components of coenzyme A, ATP. Different biomolecules of RNA and DNA are part of these things. So these can also be converted into glucose through a variety of different mechanisms as well. So if this is the open chain conformation of D-fructose, this is beta D-fructose, I want you to practice just kind of converting these and thinking about how these are oriented in space. I want you to write out the open chain conformation of D2-deoxyribose, just as a little practice problem to do. We're not going to talk about the generation of this necessarily, but different carbohydrates are also really important in blood typing. So A, B, and O blood typing has different carbohydrates that are essentially attached to different proteins in our bodies, and that is going to classify us as A, B, O, and you can kind of see these differences in the galactose, the glycanic. These are different types of sugar molecules that look different, and they determine which type we are. So that's why O blood types are commonly desired, because when we add these different groups on the top, they look a little different, and some people will reject them. So A doesn't, if you have antigens against A, then you might not be able to transfer blood from someone with A into B because they don't like this yellow carbohydrate. Where if you give O to either A, O, or B, these still are recognizable because they're shared components of these carbohydrates. So N-acetylgalactosamine, that is the previous group here, that just to kind of see what it looks like. And other uses for, besides through metabolism, that carbohydrates are used. This is heparin. It's used as an anticoagulant. It has a lot of different monosaccharides available in it. It also stabilizes various proteins and growth factors in our bodies. And this is just an example of some repeating units of heparin. They're also commonly added to different proteins to provide stability for the cell wall and to provide kind of stickiness for the cell wall. And you can see a lot of proteins will contain some short carbohydrate chains that are connected by these glycosidic bonds between the various carbon groups on the side chain of the protein with that sugar moiety here. So these can help to function as receptors or to bring different compounds in and close to the cell surface. So we're also going to look at some different types of fibers are made up of glucose or sugar backbone molecules. So this is cellulose. This is something that we cannot degrade in our body because of the orientation of these chains and how they're connected. But these generally have good interactions with each other, and you can kind of see these different these shapes. And we also, a couple of different things, we have storage of these chains depending on the type. So amylopectin is a sugar chain that, or a polymer that we find in plants, and glycogen is basically sugar branching that we see in humans, in the liver, in our muscle cells, that we can break down to access really quickly glucose for different metabolic processes. So that was a quick introduction to carbohydrates and where they are, what kinds there are, and how they can look. And now we're going to start, and we've already spoken about where we get these carbs from in our diet and how they get processed through our digestive system. Now we're going to get into some of the nuts and bolts in terms of how we get energy from those compounds. The place we're going to start here is looking at all of the different possible ways that glucose can be metabolized and where it can go into different parts of either metabolism for other things or in the metabolism of other processes. For example, we're going to look at glucose, how it gets into our cells, how it can be made into DNA components, how it can be made into glycogen, broken down to pyruvate and acetyl-CoA, where that can go into lipid metabolism or different amino acids and protein metabolisms, and also eventually go down to be further utilized for energy creation. So this is a big important picture that I'd really like you guys to know the names of the processes, what they're called, and how basically sugar or glucose is utilized throughout this process. We've spent some time already talking about glucose and how it gets into our bloodstream, and glucose is actively transported across the membrane of our cells, and once it gets across the membrane, it's actually phosphorylated generally immediately. And that phosphorylation, because this is an active transport system, that protein here will recognize glucose and allow it to come through, but then it gets trapped because now this glucose that's phosphorylated on this side is no longer recognized by this transporter enzyme, and so then it gets stuck inside. So it's the first step in the utilization of glucose as a energy molecule in our cells. Once that phosphorylated glucose, which we call glucose 6-phosphate, is trapped in our cells, there are actually a number of different things that it can be converted into. We can go through the pentose phosphate pathway, which pentose, right, 5-sugar phosphate pathway. It can be 5-carbon sugars that generate NADPH, that reducing coenzyme that we've seen, which is also can be made up of or can be utilized for RNA and DNA. That's the sugar backbone we get in those different genetic components of our cells. We can store that glucose 6-phosphate as glycogen, so long polymers of glucose, through a process called glycogenesis. We can also break that down through glycolysis into pyruvate, and we'll talk about this interaction between pyruvate and lactate. And so glycolysis can break that down. We can get some energy out of that. We can further metabolize that and get energy, break some of those carbon chains down into acetyl-CoA, and that acetyl-CoA can then be utilized to make fat, or we can be utilized to make some amino acid backbone components and proteins, or we can also take that acetyl-CoA and generate more energy in the citric acid cycle. So really big important molecules in these processes. I want you to know the metabolites at the interface for these pathways and also the name of the pathway that is going between them. And we will talk about these processes. We're going to start focusing on glycolysis first. But here's just some of the naming of those different processes, those different metabolic processes that glucose is involved in. Here's where their name kind of is derived from and what their function is. So you should be able to kind of look at this picture, but this is just a helpful chart. So these are things that you should know. These are things that are really important for this class, kind of the point of this class. But we're going to start looking at glycolysis and the metabolic processes that are involved here. Glycolysis is 10 different chemical reactions that end up breaking down glucose. You can see here we go from glucose and we're going to get pyruvate out of it. So these are the big two that you should know. You should know what glucose looks like. You should know what pyruvate looks like. And in that process, we're taking this glucose and we're going to get very oxidized carbon out of it. And In the process of oxidizing carbon, we also get reduced coenzymes, and we get some ATP out of it. So there's some energy that we can get out of this. Starting with glucose, we are going to break this down into pyruvate. There is some addition of different compounds that we are going to be looking at as well, but ultimately glucose is going to give us 2 pyruvate molecules. So what you should be able to do is look at this process and go from glucose to pyruvate and identify the different functional groups that are found on pyruvate here, and then also the chemical reactions that have to occur to allow us to go from here down to here based on the changes that are going to happen with these two compounds. I'll let you do that on your own. However, to begin, starting with glucose, we're going to walk through each of these different steps of glucose. And each step, sometimes they're even coupled with other reactions, so multiple things might be happening. The first step is that we need to trap glucose inside the cell. And we can do this by adding a phosphate group on this hydroxide molecule. So we're going to make a phosphodiester bond. And those subclasses of enzymes are called kinases. So this is a transferase reaction. We're going to transfer a phosphate group from the ATP. That's going to form an ADP, and that hydroxide is going to go onto the ADP, and then one of the phosphate groups is going to be transferred over here. So whenever we phosphorylate something, that's called a kinase. So here we can see kinase molecule. It's a hexokinase that's activating this that phosphorylation or that transferase reaction. We need to invest. There's an initial investment of 1 ATP here, and I'm going to show you on the next slide. We'll see the investment of another ATP molecule in a second. So this reaction, what essentially that first compound happens, and then this next isomerization reaction happens. So that's the glucose 6-phosphate. The second step is going to be activated by an isomerase. So the isomerization reaction, we're basically going to flip this double bond or this oxygen bond from here down to this compound, and then this carbon essentially is this carbon that's kind of coming off to this out of this chain. So that's an isomerization reaction. And then we have the fructose 6-phosphate compound here. We have a phosphorylated end here, and then the next step we're also going to again add another ATP so that we can get a phosphorylated end on this end as well. So some more energy that's used, right? This is highly exergonic, which means that we're using energy from here to here. We break that ATP bond and we form this other bond here. Or again, this is a kinase reaction. So the enzyme name is phosphofructokinase. So the phosphofructo part is that substrate, phosphofructo, and then the kinase is the enzyme, the transferase, that enzyme that's going to transfer on that phosphorylated end. Once we have our 1,6 fructose 1,6 bisphosphate, normally we number them on the right side, starting from the right side, 1, 2, 3, 4, 5, 6. So this is the 1 phosphate, that's why we get the 1 here, the 6 phosphate, 1,6 bis for 2 phosphates, and it's a fructose backbone. We break this compound into two. Essentially, we're going to cleave this in half. This compound is going to be this dihydroxyacetone phosphate, and remember this group here is going to be D-glyceraldehyde 3-phosphate. So once we have this broken in half, remember there's actually two of these groups here. We're going to have this 1, 2, 3 carbon group and then another 3 carbon group. And the difference between this and the left and right compound is where this carbonyl group is located. Right now we have, if we're looking at from the phosphate bond, it's the second carbon in where the carbonyl group is, and this compound, it's the 1, 2, third carbon in. So this is another isomerization. We don't change the number of bonds or carbons, oxygens, anything. It's just where this double bond is located. So that's an isomerase reaction. this compound here, this going from this 6-carbon chain to 3-carbon chains, that's the hydrolysis. That's where we're adding water to break apart this compound into 2 groups. Now the chemical reaction here from now on, we have 2 of these. So when we start on this next compound, I'm going to show you the rest of glycolysis goes through this compound. So we have 2 of them, but we have to convert this dihydroxyacetone phosphate into D-glyceraldehyde 3-phosphate before we move forward. So that was this was the circled compound that I had before. That's where we're going to start. And basically what happened, we used up 2 ATP already. Now what's going to happen here, we're going to, in the end, form 2 pyruvate molecules. So this is going to go through 1 time. And then the second reaction is going to kind of go through this step, right? So we're going to create 2 of these pyruvate molecules. And because this process runs through twice, we're going to make 1 ATP and 2 ATPs per pyruvate molecule that we synthesize as well. So we're going to end up with 4 ATPs from these steps, 2 pyruvate molecules, and then also the 2 NADHs because we go through that process twice as well. So let's start up at that D-glyceraldehyde 3-phosphate that I mentioned earlier. This is a coupled reaction, which means that we are both reducing a coenzyme, NADH, and we're also going to be phosphorylating this end as well. But first, in order to, what you'll notice the difference between here and here is we need to add this oxygen. So that is part of the oxidation of that carbon. We get oxidized first, and that's why this is allowed to get reduced. And then we phosphorylate it with our phosphate. So it's a coupled reaction. We can utilize, we can get that phosphate added on, and we also reduce a coenzyme at the same time, which makes it energetically favorable and allowed to happen. So we have an oxidoreductase, so there's an oxidation reaction, and there's also a transferase, so we can kind of combine those 2 enzymes, and specifically a kinase because we're adding phosphate group. Once we have this 1,3-bis phosphate, right, so we have the phosphate group on both ends, then we will use one of these phosphates to actually synthesize ATP. So now we're going to generate a energy molecule, ATP, and we eliminate that phosphate group. So now we have a phosphatase. This is another type of enzyme that's a transferase, but instead that's when we're actually taking our substrate, we're taking that phosphate group off instead of adding it. So phosphatase is kind of the inverse, the opposite of a kinase. We have this other compound, and now we are going to move. So the difference here, we're going to switch these different compounds. So it's really important that you're able to kind of recognize what is happening from one molecule to another. We're just switching these. Nothing else really changes. So it's an isomerization reaction, and it's catalyzed by the enzyme which is called, its name is phosphoglycerate mutase. So we're just moving these compounds around. We're switching those and isomerizing that compound. And then we're going to, you'll notice the next thing that's happening, we're going to eliminate this. So we're actually going to eliminate a water compound. You'll notice that that hydrogen leaves and this hydroxide leaves, and then we're left with a double bond. So this is a lyase. Whenever we generate a double bond, we can think that that's probably there's a lyase involved, usually involving double bonds. So that's a dehydrase, which is like the subclass of that lyase. But we see that double bond is formed on the bottom there, and that generates then phosphoenol pyruvate, the enzyme that catalyzes it, number 9. The enzyme's name is enolase. The next step here that we're going to use is to break this phosphate bond and actually create an ATP molecule. And we're going to transfer this double bond from this carbon up to that oxygen essentially. And we're going to have that the ketone functional group, carboxylic acid functional group is going to form. And we're going to, this is a largely exergonic reaction, highly exergonic. So it's going to we're going to use a lot of that energy to synthesize this ATP compound here, or to harness that energy. Overall, those processes, we generate 2 pyruvate molecules, 4 ATP, and 2 NADH. But in the end, remember, we generate this, but we also have to subtract some of the initial utilization in the earlier steps we used, and this isn't showing the whole thing, sorry about that, but we used 2 ATP molecules here. So we're going to have to subtract from those 4, we have to subtract the 2 that we used initially in step 1 and, or go back, in step 3, which isn't shown in that figure. It got cut off, sorry. Right, so step 3, there's also ATP used. So we actually, the net output is 2 ATP, 2 NADH, and 2 pyruvate molecules. I would like to point out that there are other entry points for different sugars into glycolysis. For example, fructose here. There are multiple places that can enter. In the muscle, it's phosphorylated into fructose 6-phosphate, and it goes directly into step 2. In the liver, it's converted into glyceraldehyde 3-phosphate, which is part of step 5. So that's where it's broken down already. So it just depends on where it's coming in. But all of these different fructose, galactose, mannose, we have enzymes that convert them and make them into either fructose or glucose so that they can enter in different regions in the pathway. So, different fruit sugars like fructose or milk, galactose is a sugar from milk, mannose we can get from beans and different kind of legumes. So we can get sugars into our diet in different ways and into this pathway so we can generate energy. And in addition, pyruvate, the reason I want you to know this compound is because it's a linchpin, it's a center molecule, right? It's a product of glycolysis, but we can also use it to make different, so oxaloacetate and alanine. Alanine is an amino acid. Oxaloacetate is also something we can generate amino acids from. It's also amino acids or proteins, groups, all we have to do is add nitrogen on here and we can get amino acids. We can go into acetyl-CoA. So this is also links us to fat. Different compounds, for example, in yeast we can make ethanol. In other mammals and animals we make lactic acid or lactate. So this is a kind of a central molecule for a lot of different metabolic processes and links to the things that we have to make in our body, different proteins, amino acids. Right? We talked about how glucose. So pyruvate's the central molecule. It's really important for a lot of different needs in our body. I also mentioned pyruvate can be converted into ethyl alcohol. This can happen in yeast. Yeast do this pretty easily under anaerobic conditions. So when there's not oxygen available to it, it will actually synthesize and make ethanol. Remember, we get some energy out of this. So if there's sugar available, we can break that sugar down into pyruvate. But then if you build up too much pyruvate, that's not good either. So it needs to kind of get rid of this pyruvate and turn it into something else, so that final chemical reaction that synthesizes pyruvate can occur and we can still make some ATP out of it. So that we can change into ethanol. In yeast and in humans, we make lactic acid. So the lactate is right here, shown here, and that is our final product that we can make if we don't have oxygen, if we're running low on oxygen. You've heard of lactic acid buildup. That's the buildup of lactic acid when we don't have oxygen to then these next steps in order to make acetyl-CoA and go into the citric acid cycle. We need, for that process, we need oxygen. So if we don't have oxygen, we can convert that pyruvate to lactate. And that's then taken care of. We can take that lactate here. This is called the Cori cycle. Cori cycle, and actually we take that lactate and it can then get transported in our blood to our liver. And the liver cell can take that up. It converts the lactate into pyruvate.
and we can do gluconeogenesis. That's the next step we're going to look at. We're going to talk about gluconeogenesis. A lot of this happens in the liver, and your liver, its primary function is to regulate glucose levels in your bloodstream. So when you need to take lactate up and transfer this back so you can get glucose back in your system, then your muscle cells can have more energy, more glucose. Even if you're low on oxygen, you can't make any more energy from the pyruvate, but you can have your liver actually put energy in and make more glucose so that you can get energy out for your muscles to function properly. So you can look at these and determine if the pyruvate's being oxidized or reduced in these situations, but I'll let you do that on your own time. And you can pause or unpause it here. In aerobic conditions, when we have enough oxygen, this pyruvate, we're going to lose this carbon dioxide molecule, and it's going to continue down into the citric acid cycle. So we have this CO2 leaving, and then we're actually going to also bind on this acetyl S-CoA. We're going to have a covalent bond actually being formed. This is an acetyl group that you'll see in the future. So the acetyl group, that's why it's called acetyl S-coenzyme A. So this pyruvate molecule will diffuse across the mitochondrial membrane. So most of this, the glycolysis, is happening in the cytosol of any cell. This pyruvate gets transported across into the inner mitochondrial membrane, and then this pyruvate dehydrogenase is involved in this step here. And so we'll talk about the glycolysis, or sorry, the citric acid cycle later on, but this will be the starting point with the acetyl S-CoA. So again, a question I'll let you
answer on your own time.
Once we're going to go back to this kind of idea of the Cori cycle. The gluconeogenesis, so glucose we're trying to make the creation of new glucose. So this is called gluconeogenesis because we're creating new glucose. And this largely is happening in the liver. So the gluconeogenesis a lot of time happens in the liver. One of the starting points for this process is lactate. Otherwise, we can also use pyruvate. But a lot of times our liver is taking this up from the blood, from leftovers that if we make lactate, if we don't have enough oxygen available to continue on with the rest of the carboxylic acids or the citric acid cycle. So glucose is the primary food for most cells in our bodies. Most of this gluconeogenesis, again, is happening in the liver. Ultimately, what we're going to do is we're going to go backwards. We're going to take 2 pyruvate molecules, and we're going to make 1 glucose molecule. So we'll talk about some of the energetics and how many of these different molecules we make at the end of this section here. But if we're going to compare glycolysis with gluconeogenesis, there are different steps that are going to be really important. So this step is one step. We're going to have another step here and another step here. So when we're breaking these down, this first step, these take they use a lot of energy. There are big energetic steps here that are hard to be reversible. So they're unreversible steps when we're actually using ATP or synthesizing ATP. A lot of times that energy between those 2 compounds are too great to just have one chemical reaction occur. So these are 3 reversible steps that we actually use different enzymes to actually climb back up that pathway here. So when we start with this pyruvate molecule, we actually use 2 steps to get it back. So you can see pyruvate to phosphoenol pyruvate. We actually use 2 enzymes to get back up to that initial compound. The first step we do is we take ATP, and we're going to then use that. We're going to use that biotin. We're going to couple that reaction with ATP, and we're going to carboxylate this reaction. Usually biotin is used in these carboxylase reactions. It's usually a common coenzyme for those types of reactions. So we use 1 ATP, and we have this carboxylated enzyme. And the second step also requires the use of now another ATP or GTP. ATP is usually used in plants. GTP is used in mammalian cells or animals. And we're going to phosphorylate this carbon, and we're going to generate the phosphoenol pyruvate, and then we've used up another ATP molecule. So the commitment, we've just used 2 ATP molecules right off the bat. So this suggests that we have really decided in our cells that we're going to commit to making glucose. That's why all of our cells don't typically do this. That's why our liver is very valuable, because it can make a decision, and it's going to switch all of these enzymes around. It's going to create these phosphoenol pyruvate carboxykinases, PepCK. It's going to upregulate these and make more of these and invest in this this process for going back up the pathway. So we've created that phosphoenol pyruvate. Everything else then flows depending on the concentrations. So if the concentration increases for phosphoenol pyruvate, all of those steps are going to flow up until we get to this next irreversible step. So remember, we have these two compounds. We have the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate. We need one of each of those. So we create one of these, and then we have to kind of glue those back together. We have to put those back together. So we have this fructose 1,6-bisphosphate. But all we do then is we lose a inorganic phosphate. Here, going down glycolysis, we actually use an ATP molecule. Once we're going back up, we don't resynthesize this. So this is where we kind of lose some energy here. We lose some high energy molecules. So we simply have that inorganic phosphate leaving, and then we generate a hydroxide. We make that alcohol. Same thing happens for this last step. Once we lose this glucose 6-phosphate, the enzyme's a phosphatase, right? We lose that phosphate group, and we go to glucose. So here we go, we lose this phosphate group, and then we have this glucose molecule that's left over. Why do I have this chain breaking? Well, this is what's kind of holding this phosphorylation is what's really holding that glucose molecule inside the cell. Once this looks like glucose, that transporter that we talked about earlier can recognize the glucose and allow it to release. Especially if this is happening in our liver, it's pumping out a ton of this glucose molecule all at once. So we should have a buildup of concentration of glucose. And then in our blood, usually when our liver is making glucose, there are low glucose concentrations in our blood. So there's high probability that the flow of glucose is going to go out of the cell rather than into the cell in that instance. So if we compare these things, you should be able to look at and count up how many different ATPs are being synthesized when we go down during glycolysis, and how many ATPs we use up when we go up to gluconeogenesis. So you should be able to do that math and figure out the difference there between those two different pathways. So I do want to talk about some of this process with gluconeogenesis, kind of a case study here. Some of the research that's been done looking at different enzymes within this pathway of gluconeogenesis. A group has looked at, if you look at the wild-type mouse, I'm going to show you a video in a second. In the back is going to be a wild-type mouse, and in the front it's going to be a mouse where we've upregulated this phosphoenol pyruvate kinase in the liver. So there's more of this in normal cells, in this mutant mouse than there are in normal cells. So you can see here, this super mouse, it has the PepCK mutant. This is the older mouse, the control mouse. It's going to run for a little bit, but you're going to see eventually it's going to just give up. So this is 0.2 miles. And that one's just, yeah, I'm done. So this keeps going for an hour. It's still running an hour later, and it just kind of keeps going and going and going. 3 hours later, and the total distance, it almost runs 5 kilometers after this, or maybe a little more, right? It's still kind of interested in running on here. So it's a busy guy. The mouse in that video simply had a change in this one enzyme, and it allowed it to do some crazy things. There's not really a biochemical explanation for the hyperactivity. It's likely leaner because the liver can break down fat easier as well. So we can use pyruvate. Remember I mentioned that's a link between pyruvate and the ability and fat. So it's likely that this pathway can also be utilized to break down and utilize fat for energy from our liver as well. And so they tend to be a little more hyperactive, a little more aggressive, generally more lean, which kind of makes sense a little bit as well. So based on what you know about gluconeogenesis, where would you predict or guess that this enzyme would normally be expressed? And where do you think, how would you describe a detailed experiment that would examine where this protein is and how it's expressed? How would you figure this out? What kind of assay would you run on this mouse to figure out what's actually happening? So that's something that that you should be able to do based on some of the information that we've talked about in this class so far. As I alluded to before, that super mouse, right? We had mentioned that we might be able to use fats a little more easily than others because here there's that link again between fat and pyruvate. And so fatty acids can be used to make pyruvate, then that can go up to make glucose. That's one pathway. So a lot of different precursors for gluconeogenesis, not only through fat, but different proteins, amino acids, lactate, and oxaloacid or different amino acids can lead into their fats from different pathways, not only from triacylglycerols, but from this end as well. So a lot of different components here. Acetate here, these are different, not generally we don't take CO2 up or acetate up to make acetyl-CoA. Some bacteria and single-celled eukaryotes can. Propionate is another one that humans don't do, but for example, cattle can. This seems like a good spot to stop, and the next part that we're going to talk about is in how we can regulate some of these enzyme pathways.